So, this is the part 2 of the class of the digestive system, the nutrition of human beings. So now we are having clear idea of the human digestive tract, and we know what food we are going to take and their ultimate products. Now relate them. When you are going to take the food, teeth are there and the tongue is there. There will be the glands. So what are the glands we are having? Now let's start regarding the knowledge about the glands. To intake the knowledge about the glands. First, salivary glands are there. Okay. Then there will be, when we are going to the stomach, just there related to that particular region, above the stomach there is the largest gland of human body, liver, and there will be another very important part, that's the gallbladder, and gallbladder is connected to the liver. Liver will secrete the bile juice. That bile is coming to the gallbladder. Okay, and again gallbladder is connected with the pancreas, another gland. Okay, so hepatopancreatic duct, and it will release its content to the first part of the small intestine. So now name me the first part of the small intestine, the duodenum. In duodenum, bile will come from pancreas, pancreatic juice will come through which duct? Hepatopancreatic duct. Okay, fine. So now, foods you have taken, suppose salivary gland release the salivary amylase, first enzyme. Salivary amylase will work in the alkaline medium. In your mouth, here, pH is about 6.8. So it's a basic alkaline, okay. Now, here salivary amylase will cleave the starch or the carbohydrate things to maltose. So first product, carbohydrate, is broken to the maltose. Fine. So maltose is here formed. Protein, carbohydrate, means protein, lipid, not properly digested here. Mastication of the food done here, and here is the formation of bolus, bolus. This bolus is now passing through esophagus. Now, an interesting fact. The movement of your this mouth to foot, this foot pipe, is not gravity related. Means if you are in the inverted position, upside hanged, then also if you are going to take the food, your food will go to the esophagus. It will not come outside. How? There is a particular movement. Peristalsis. Peristalsis is the movement of your esophageal muscles. So it's not like that you are going to take the food and it is just dropped in the stomach. No. The particular movement of the esophagus is known as the peristalsis, and that will be performed through that food will go to your stomach. Not like directly. Okay. Now, when food is going to the stomach, there another set of digestion started. What way? Here in stomach, 3 kinds of cells are there. First, mucus cell, mucus neck cell, they will secrete mucus. Next, chip cell, main cell, they will secrete pepsinogen. Third, another set of cell, they will secrete the HCl. I am not going to take all the names, chip cell, parietal cell, so many, in brief. So first thing is mucus cell, second thing is pepsinogen from chip cell, and third one is their secretion of HCl. Now what is the role of mucus? Each and every day your stomach is releasing about 2 liters of HCl. 2 liters of HCl. That means it's highly acidic. pH 1.8. So your stomach cell lining wall is damaged. So to protect it, mucus lining is there. Okay. Now, why stomach is releasing HCl? Unnecessary? Of course not. Pepsinogen is secreted, but it's the precursor form of the pepsin. It can't work. It's inactive zymogen, or we can say inactive enzyme. Okay. So this inactive enzyme, pepsinogen, in presence of HCl at the pH 1.8, cleave to pepsin. Now a bit clear. Pepsin is going to work on carbohydrate, fat, no, protein. And now you say, if proteins are broken, what will be the product? Amino acid? No. The intermediate forms, proteose, peptone, directly not amino acid. Okay. So now recap. Pepsinogen cleave to pepsin with the help of HCl. Okay. Now pepsin will break the protein to this proteose or these peptides. Okay. Done? No? Okay, fine. Apart from this, there will be a few lipid breaking enzyme. As now already we are a bit accustomed with the enzyme name. Generally the enzyme names are having ASE, ASE, like protein breaking enzyme, protease, lipid breaking enzyme, lipase, sucrose breaking enzyme, sucrase, maltose breaking enzyme, maltase. So everything you can read out with an if. It's very easy if you are reading with love and care. So now next thing, a bit major thing, protein digestion, nothing more. Here another after that, in small intestine the food will move, and there who is present? Small intestine first part, duodenum. Here connection of the hepatopancreatic duct. So here first time lipid digestion properly started. In hepatopancreatic duct, therefrom you can remember hepatic. Hepatic means liver. Therefrom connection with the gallbladder. Liver will release the bile. Bile is stored in the gallbladder. From gallbladder it is directly coming to the duodenum. Here from the pancreas also pancreatic juice is coming to the duodenum. So one by one I will first describe what is the role of bile, then pancreatic juice, then duodenum oriented intestinal juice. First, bile. Bile does not contain any enzyme. So why important? Very important. Fats cannot be broken directly. It needs the emulsification action. Emulsification means broken into smaller droplets. Name is micelle. MICELLES, micelles. Fats are broken into micelles with the help of this particular bile. Okay. Now, next important action. In small intestine, for any enzymatic action, we need the alkaline medium. Food coming from the stomach is highly acidic. So, to neutralize, bile added. This adding some salt to make it alkaline. So two functions, hopefully you understood. Second, you are moving to this part. Means which part? Pancreas. Pancreas is releasing pancreatic juice. Here almost 3 enzymes are present. Means protein breaking, fat breaking, all these things. Protein breaking enzyme, most important, trypsin. Some others are also there, like chymotrypsin, carboxypeptidase. At class 10 level, you must remember at least trypsin name. Trypsin is formed in the precursor form trypsinogen. Trypsinogen here, which will form the trypsin in duodenum. Sorry. And other things are chymotrypsin, carboxypeptidase for protein breaking purpose. Secondly, here also we are getting the pancreatic lipase for fat breaking. These things here we are getting. Now coming to the next part, in the intestine. Intestine, here we are getting the intestinal juice. So bile is acting these pancreatic juices are also acting, and here this intestinal juice is also acting. Intestinal juice name is succus entericus. The succus entericus or the intestinal juice, it is contained of many protein breaking enzyme like peptidase or dipeptidase. Also here the lipase enzyme. Also here the sucrase, means sucrose breaking enzyme. These things all are here. Fine. So ultimately they will break to the ultimate product, means the monosaccharide and the amino acid and the fatty acid glycerol. Okay. If I say it verbally, you may face problem. That's why I will show you in the next video what are the products. Ultimately, small intestine, this duodenum part for the digestion purpose over jejunum and ileum, especially ileum, the means finger like villi for the absorption purpose. Okay. After that large intestine, that's totally for the absorption of water, minerals, some medicine, ultimately fecal material formation. That's the overall idea of the digestive system. Now in the next video I will show you regarding this path or diagrams. Okay.